Welcome back to Peking University MOOC, Bioinformatics, Introduction and Methods. Let's continue with our lectures on ontology and identification of molecular pathways. In the first two units, we took a close look at the gene ontology and the KEGG pathway database. However, their nice hierarchical vocabularies wouldn't be very useful if the terms were not associated with genes and gene products. How were the genes and gene products associated, or sometimes called annotated, with geoterms and KEGG pathways? How would you do it? Please pause for a minute to think about this yourself before continuing. The Gene Ontology Consortium uses three ways to associate or annotate a gene with a geoterm: annotation through manually reviewed experimental evidence that links the gene with the geo function, annotation through manually reviewed computational analysis evidence that links the gene with the geo function, annotation by completely electronically generated computational analysis evidence to link the gene with the geo function. Let's look at each type of annotations more closely. Direct experimental evidence that had been manually reviewed by experts at the Gene Ontology Consortium, often called curators, is usually considered the strongest evidence. The curators review the published literature for experimental evidence linking genes and gene products to molecular functions, biological processes, and cellular components. There are several types of manual annotations by experimental evidence, including IDA, inferred from direct assays such as enzymatic assays, in vitro reconstitution, immunofluorescence, cell fractionation, and physical interaction binding assay. IPI, inferred from physical interaction, such as two hybrid interactions, co-purification, co-immunoprecipitation, and iron protein binding experiments. IMP, inferred from mutant phenotype, meaning experimental studies of mutations, polymorphisms, or allelic variations, any procedure that disturbs the expression or function of the gene, overexpression or ectopic expression of wild-type or mutant gene, and so on. IGI, inferred from genetic interaction, such as traditional genetic interactions, functional complementation, rescue experiments, and interference from a different gene. IEP, inferred from expression patterns, such as transcriptional levels or timing and protein expression levels. And EXP, inferred from experiments that are not covered in the above list. Unfortunately, direct experimental evidence is only available for a limited number of genes and gene products. Because many genes are conserved across different species, predictions of the molecular function, biological process, and cellular component of a gene in a less experimentally tractable organism can sometimes be made based on sequence and structural similarity to a well-studied gene in a well-studied model organism that already has a geo-annotation. A curator can manually review the computational predictions and copy the geo-annotations of the well-studied gene to the new gene. These annotations include ISO, inferred from sequence orthology, either experimentally or phylogenetically, to an orthologous gene that already has a geo-annotation. ISA, inferred from sequence alignment by a pairwise or multiple alignment tool such as BLAST or CLUSTAL-W. ISM, inferred from sequence model by prediction methods for protein domains, protein features, motifs, and non-coding RNAs. ISS, inferred from sequence or structure similarity not covered above. Other annotations include IGC, inferred from genomic context, such as operon structure, syntenic regions, pathway analysis, and genome scale analysis of processes. IBA, inferred from biological aspect of ancestor by phylogenetic evidence. There is also an IBD inferred from biological aspect of descendant, which is defined in GO, but for some reason no gene is associated with it. Next, we have RCA, inferred from reviewed computational analysis. This is different from the above in that this refers specifically to analysis of large-scale experimental datasets or integration of datasets of several types. The last two are negative evidence and are annotated as not in gene ontology, including IKR, inferred from key residues for a gene product which, although homologous to a particular protein family, had lost essential residues, and IRD, inferred from rapid divergence, where there exists phylogenetic evidence of rapid divergence from ancestral sequence. Now, I'd like you to pause for a minute, look at the whole list on the page again, and try to remember which of the methods you have learned in this MOOC so far can be applied here. Finally, because the speed at which genes and genomes are sequenced far exceeds the speed at which experimental biologists can study them in the lab, or the speed at which any curator teams can review the evidence, many genes were annotated by completely electronically generated computational analysis without manual review. These are usually considered weaker evidence than manually reviewed evidence, but they are invaluable in covering order of magnitude more genes. They are labeled as IEA for inferred from electronic annotation. The methods used for the inference are similar to what we saw in the last slide. There are some annotations in gene ontology that don't have direct experimental data and associated with it, but are mentioned in review articles or the discussion section of research articles. They're marked with TAS for traceable author statement if the statement is justified with a citation, or NAS for non-traceable author statement if the statement is not justified with a citation. In addition, there is IC for inferred by curator without specifying the source of evidence, and D for no biological data available, and NR for not recorded. You may want to be cautious about using these annotations and try to dig out the direct supporting evidence yourself. So, among all the genes annotated in gene ontology, what percentage of the annotations are based on experimental evidence, what percentage are manually reviewed computational predictions, and what percentage by completely computationally predicted evidence without manual review? What would you guess? Well, it depends on the species. Human and famous model organisms such as mouse and Drosophila tend to have more annotations based on experimental evidence and manual review of computational predictions, whereas non-model organisms whose genomes were sequenced tend to have most annotations based on purely computational predictions without manual review. For instance, as shown in this figure, among all fruit fly genes, 40% were annotated by experimental evidence, 20% by manually reviewed computational evidence, 20% by other author or curator statements, and only 20% by computational evidence without manual review. By contrast, for genes in the pig genome, 90% of the annotations were based on computational evidence without manual review. Annotations in the KEGG pathway database are done in similar fashions with a combination of manual curation and automated analysis based on a combination of experimental evidence and computational predictions. So, After the good people at the Gene Ontology Consortium and the Pathway databases had created the hierarchical vocabulary and associated lots of genes and gene products to them, you, as users, can now use these resources to analyze genes of interest from your own research. What analysis can you do, and how can you do them are the topics of the next unit. I look forward to seeing you then.